Now we're going to talk about what happens in the proximal convoluted tubule and the distal convoluted tubule. Again, in the proximal convoluted tubule, it's mainly reabsorption. So in the glomerulus, we filtered, so we pushed everything out. Now we're going to reclaim most of what we pushed out. So again, never urinating glucose as all normal, all is well. Not urinating out amino acids, specifically ones that we need, and all of our other organic nutrients like lipids reabsorbed. We want to keep those. We can use those inside the cells to build new things or use them to make ATP. The proximal convoluted tubule, again, is going to resorb a majority of sodium, potassium. It's also going to reabsorb bicarbonate. That's our major buffer in the bloodstream, so we want to keep that. You'll maintain magnesium, phosphate, sulfate, ions. All of those things are going to be reabsorbed. Again, most of the water, 60 to 70% of the water, going to be reabsorbed by the proximal convoluted tubule. So what we can see is as reabsorption occurs, so as you're going down the proximal convoluted tubule, solutes are leaving. So these glucose, amino acids, all these ions are all leaving and going into the peritubular space. That helps with water reabsorption because you're going for all the solutes are out here and water wants to go hang out with its solute friends. So it's going to leave as well. So that's going to help again with our water reabsorption by just making an osmotic gradient. So again, this is in the proximal convoluted tubule. So this is in the lumen tubular fluid. This is what has been filtered out. So this is our filtrate here. And what's happening in the proximal convoluted tubule? Reabsorption. Other organic solutes are going through the cells of the proximal convoluted tubule, going into the peritubular fluid, and then diffusing into the peritubular capillary. Ions, so all the ions that we talked about, so they're going to be absorbed. They can happen with diffusion, so high concentration to low concentration. Facilitated diffusion, we have a carrier or channel, but it's still going from high to low concentration. Or we can have the two types of active transport, active, just regular active, or secondary active transport. So there are many different ways we're going to get ions reabsorbed based on what they are. I'm not going to go over individual how they are, but many ways that we're going to bring ions from the filtrate back in the cells to the peritubular fluid to the capillary. Because of glucose, solute, and other organic solutes and ions are going into the peritubular fluid, water says, hey, wait for me. So osmotic water flow is going to happen because of the movement of solutes. So water is going to follow all these solutes and go back into the peritubular capillary and be reabsorbed that way. In the distal convoluted tubule, so we skip the nephron loop, which we'll talk about next or a little later. So reabsorption and secretion kind of linked. So by the time we've gone through the distal or the proximal convoluted tubule and the nephron loop, about 15 to 20% of what we initially started with is going to come to the distal convoluted tubule. So we've gotten to a point where filtrate is very different from what we started with. So remember, we started with the same as plasma, and we've changed different concentrations of solutes until we get to the distal convoluted tubule. So we can look at hormonal control of the distal convoluted tubule. One of them is going to be aldosterone. When the presence of aldosterone, more sodium is going to be reabsorbed. So you're going to take in more sodium. And because we're reabsorbing sodium, we have to exchange it for another positive ion, and so we get rid of potassium. Just kind of oppose it. But the major one is sodium, and then water will follow. So remember, aldosterone is going to increase blood volume, which will increase blood pressure by sodium retention or reabsorption, keeping in the salt. We can also have hydrogen ions that are secreted, meaning they're put into the tubular fluid. They're removed from the peritubular capillaries and moved into the tubular fluid. And that can also exchange for sodium ions as well. So sodium ions reabsorbed and hydrogen ions taken out. So this is getting rid of hydrogen ions. That means your blood is at a low pH. There's too many hydrogen ions. We have too many. So how do we need to increase the pH back? Get rid of hydrogen ions, and the kidneys are where to do it. We also have secretion of certain toxins or drugs that we need to get rid of. So carrier proteins in the distal convoluted tubule are going to take toxins and bloods from the peritubular fluid and put it into the tubular fluid to get rid of it. So looking at the picture again, this is the distal convoluted tubule. So same thing that we saw before. We have the tubular fluid filtrate here. We have the peritubular fluid, and then we have the peritubular capillary. So if aldosterone's around, sodium ions are going to be reabsorbed, meaning they're going to be taken from the tubular fluid and brought back eventually into the peritubular capillary. And to exchange it, we're actually going to get rid of potassium. And that's going to, again, we'll have water follow. So that's going to increase water reabsorption too. So water's going to be in here. If you have a low pH in body fluids for whatever reason that's occurring, then hydrogen ions are going to go the opposite direction of sodium. So hydrogen ions are going to be removed from the capillaries, go into the cells, and be released into the tubular fluid. Sodium is going to go opposite because we have to make sure our charges are equal. So we have another cation that's going to come in, and we like sodium because then water will follow. Again, we have carrier proteins that are going to help get rid of toxins and drugs. If they kind of avoid going into the filtrate at the glomerulus for whatever reason, if they're able to avoid it, we can pick them up here. And so we pick them up and move them out with carrier proteins into the tubular fluid as well. So now looking at the nephron loop. So we've kind of talked about the convoluted tubules. Now we're going to talk about the one in between those. So remember, it's made up of a descending limb, so a limb going down, and then it makes a U-turn, and then there's an ascending limb going up. The permeability of these sides are different based on whether you're doing it. So when you are going down, only water can leave the descending limb of the nephron loop. So only water can be reabsorbed here or moved here. Solutes cannot pass. So they are staying in. In the ascending limb, it's the opposite. Water cannot leave, it's stuck. Because water has to have channels. They have to have aquaporins. So there's aquaporins on this side, nothing of aquaporins on this side. And most of the time, solutes that we're talking about is going to be sodium and chloride, but we also have some potassium and some hydrogen as well. But it's mainly going to be sodium and chloride. So when we are in the nephron loop, we have what we call countercurrent multiplication. So what happens is we have that U-turn. So tubular fluid, we have kind of the U-turn. Tubular fluid's going down in the descending limb towards the renal pelvis. That's what it means there. So renal pelvis. And then when we're in the ascending, it's going in the ascending direction of going towards the renal cortex. So that's the countercurrent. They're going in opposite directions, countering each other. So as the multiplication, the actual effect of the solute and fluid, the fact that water can leave here, so the solute concentration of the fluid in the tubule is going to be highest down here, because as you're flowing and you make the U-turn and go around, water's left. Because remember, water can flow out of the descending limb. So you're going to have the highest amount of solute here. Then as you're going up the ascending limb, sodium and chloride can leave. So you're going to decrease your solute concentration again. But in the paratubular fluid, you're creating an osmotic gradient. So adding sodium chloride to the paratubular fluid makes, on this side, more water want to leave, because it wants to follow sodium mainly. So that's what we get with the countercurrent multiplication, is kind of we're going in opposite direction, and as we're kind of going through, the effect is going to be stronger. We're going to be able to absorb more water because we're bringing out more sodium, and water is going to follow sodium. We create that osmotic gradient. This is important. Again, this is going to be most of your water reabsorption is going to be in the proximal convoluted tubule, but 20 to 25% is going to be in this area. So we're making even concentrated urine. If we need to even more, this is going to come into more play in our juxtamedullary. So we have a longer way, so the loop is a lot longer. So just more time. More water, more water, more water, more solutes to make the water come out. So you have even more time for water to be reabsorbed. The more water that's reabsorbed, the more concentrated the urine's going to be. So again, we're going to look at this. So in the ascending, so again, we're going up towards the cortex. That is going to be where solutes can leave. So again, it's impermeable to water. Water can't leave. Water must stay. But we actively transport sodium and chloride out of the tubular fluid into the paratubular fluid. So the fluid surrounding the nephron loop. So you're decreasing the tubular fluid solute concentration, but you're increasing the paratubular fluid concentration. Because again, this is going to create an osmotic gradient. So water is going to want to leave the descending limb. So again, osmotic concentration of the paratubular fluid is increased because of the ascending limb. And so now we're in the descending limb. So we kind of got opposite. So this is the first part from the proximal convoluted tubule. You go down. So this is going medulla. So this is permeable to water, meaning it allows water to move. And it doesn't allow any solutes to move. So nothing's transported with the sodium and chloride. But because water is allowed to move and there's an increase in sodium and chloride concentration in the peritubular fluid, water wants to leave because it wants to follow the solute concentration. So we get to the point where we're going to have more water reabsorption because water is going to move from the tubular fluid to the peritubular fluid because of osmosis. There's a higher concentration of solutes out here, and so water is going to want to move. Again, as water is leaving, the highest solute concentration of the tubular fluid is going to be at the bottom. And then as you're getting get rid of more sodium and chloride, you're going to decrease the solute concentration. So you're going to get to the point where you're going to increase it and then you're going to decrease it again. So again, only water can move out of the descending limb of the nephron. Only solutes can move out of the ascending limb. Just making that osmotic gradient, because what does the nephron loop do? Water reabsorption. So it's going to increase urine concentration. Less water. So again, as fluid's moving the descending limb, water moves out. Again, because of the concentration gradient that's in the peritubular fluid. There's more solutes out here and less water. Water is going to move from high concentration to low concentration. So it's going to want to move out. It follows solutes, so it follows sodium. So it's going to want to leave as much as possible until you get to a point where it changes over where it can't leave. And so this side is making that osmotic gradient. And that's why we have the counter-current exchange. So again, it's going down this way, up this way, and you're concentrating your urine. So again, as fluid's going up the ascending limb, sodium and chloride or chlorine is going to move out, and they're actively transported. So this isn't because of diffusion. You're going to use energy. You're going to use ATP to pump those ions out of the tubular fluid into the peritubular fluid. So you're going to move it out actively, pushing it out. Because again, the goal is to make that osmotic gradient to make water want to leave on this side where it can. So the concentration, so water is still also reabsorbed against along the distal convoluted tubule and the collecting ducts. This can cause the increase of concentrate of solute. So here you go. So it can increase the solute concentration in the tubular fluid because water is going to continue to be reabsorbed. So we've kind of gotten to the point where we got rid of a bunch of solutes with the sodium. Well, then all of a sudden we're going to increase it again because we're going to allow water reabsorption in other areas as well. So loss of water is going to increase the concentration of solutes because the water's not diluting it anymore. So the distal convoluted tubule and the collecting ducts are impermeable to urea. So urea actually can't leave. So that's going to maintain the solute concentration inside the tubular fluid. Plus you want to get rid of urea. It's a waste product. And they have what we call variable permeability to water. Why? Because we can change it. It's not always the same. It's the same in the proximal convoluted tubule, and it's the same in the nephron loop. So we look at two types of water reabsorption. So first, we're going to have most of it is going to be tubular reabsorption of the water is due to that movement of sodium, mainly other solutes, to create that osmotic gradient. Again, water is going to want to move from high concentration to low concentration. That makes osmosis, and usually it's a facilitated process because you have channels, water channels or water pores that are called aquaporins. It allows more water to move through the cells because remember, water needs a little help because it is a polar molecule, so moving through cell membranes is a little more difficult. So we have two types of water reabsorption. Both happen because of osmosis, but we have obligatory water reabsorption and facultative water reabsorption. Obligatory is you're obligated, meaning it never changes. Aquaporins are always in the proximal convoluted tubule. Never add or subtract them. They're always there. That's why, again, majority of water is going to be reabsorbed in the proximal convoluted tubule. Facultative water reabsorption is you can add aquaporins if you want to. So you can actually add them to the collecting ducts in the distal convoluted tubule if your posterior pituitary releases ADH. So antidiuretic. So again, hormone, it's the anti-P. So you want to decrease your urine volume, which is increase the concentration of your urine. So how do you do that? You increase your water reabsorption. So ADH is around, causes more aquaporins to be around. More aquaporins means more water able to move. So again, the water permeability of the proximal convoluted tubule and the nephron loop can't change. Can't be a change. It's obligatory. It's obligated. You can't change your obligations. Water will always move because there's always a channel. There's always aquaporins there as long as there's an osmotic concentration gradient. So as long as there's a reason for water to move, it's going to move. So again, that's obligatory water reabsorption. We can change it in the distal convoluted tubule and the collecting duct. Again, that's facultative water reabsorption because we can change it based on what we want to do. So again, ADH or the antidiuretic hormone is going to have insertion of aquaporins. If you insert aquaporins, you're going to have more water reabsorption. So this is looking at a normal, kind of not having ADH. So this is no ADH around. This is where you don't need it. So this is showing you proximal convoluted tubule will have water reabsorption. You go to the nephron loop. So the descending limb, you have all this water moving in because of the osmotic gradient. And then in the ascending, you have creating that osmotic gradient with sodium and chloride active transport. And then nothing, no water reabsorption in the distal convoluted tubule or the collecting duct because you don't have aquaporins. So if you are not increasing your water reabsorption, you're actually going to have more dilute urine. So you have a larger volume and more dilute. You don't need to reabsorb more water in these areas. And your brain or your posterior pituitary is not telling you to do so with the hormone. Then you see the same thing that's happening here. Nothing's changed over here, nothing's changed over here. But now you have ADH involved. So ADH causes aquaporins to be added to the distal convoluted tubule and the collecting duct. And so then you're absorbing more water than you would. You're allowing water movement in these areas. So look at all the solute concentration. The solute concentration is going to increase significantly. So you're going to get a small volume of concentrated urine because of ADH. Remember, it's the anti-P. You want a small amount of concentrated urine.